The last session before break time is development of new SARS-CoV-2 therapeutics. For that, I'd like to invite the two session chairs, Monica Herrera and George Carlin Newman of Bio-Rad Laboratories.
Thank you, Tal. Good morning, good afternoon from wherever you may be joining us. Welcome to this session, Development of SARS-CoV-2 Therapeutics. I am Monica Herrera, Director of Molecular Diagnostics at Bio-Rad, and I'm joined by my colleague, Dr. George Carlin Newman, Director of Scientific Affairs. Let's begin. Our speakers today will discuss four broad topics covering the spectrum of therapeutic options for COVID-19. Our discussions will be divided into four mini sessions, each comprised of two of our experts who will share a brief presentation followed by a short Q&A. Dr. Stephen Quay and Dr. Ashish Manglik will focus on prophylaxis. Dr. Daniel Swaney and Dr. Anu Osinusi will discuss antivirals. Dr. Aaron Tobian and Dr. Peter Marks will talk about convalescent plasma. And we'll end with Dr. Maximilian Koenig's and Dr. Bruce Patterson's talk on anti-inflammatories. I will introduce each speaker before their respective session. But first, I would like to frame the discussion and underscore why COVID-19 therapeutics is more relevant than ever. Although at the end of 2020, we had remarkable good news with the Pfizer, BioNTech, and Moderna vaccines being authorized for use and others following in the pipeline, we are a year into this pandemic and global cases now reach over 95 million, with over 2 million deaths and counting. We need more research and development, more manufacturing, and greater access to novel and repurposed compounds to treat the disease as we gain further understanding of its pathogenesis. Most people who contract COVID-19 experience only mild symptoms, but clinical deterioration is described in about 15 to 25% of reported cases. The pathology of COVID-19 includes direct damage by the virus, as well as damage from a hyperinflammatory response. As we can see in the figure, when the immune system goes into overdrive, inflammation can spread throughout the circulatory system, leading to a cytokine storm. This systemic inflammation can cause septic shock, where blood pressure drops dangerously low and organs can no longer be perfused, leading to multi-organ failure and even death. And so it is two main processes that seem to drive the pathogenesis of COVID-19. Early in the course of the infection, the disease is primarily driven by replication of SARS-CoV-2. Later in the course of the infection, the disease is driven by an exaggerated immune and inflammatory response to the virus that leads to tissue damage. Based on this understanding, it is anticipated that antiviral therapies, which prevent the virus from multiplying, would have the greatest effect early in the course of the disease, while immune modulators or anti-inflammatory therapies, which help the immune system to fight the virus or stop it from overreacting dangerously, are likely to be more beneficial in the later stages of COVID-19. And some potential therapies may act in different ways or via multiple mechanisms. And there are researchers around the world, and we have 8 of them as panelists in the session, working at record speed to find the best ways to treat and prevent COVID-19, from investigating the possibility of repurposing existing drugs to searching for novel therapies against the virus. There are all sorts of ongoing studies focusing on the spectrum of clinical presentation and potential pharmacological treatment. As of January 17th, there were over 2,200 ongoing clinical trials, and the number of completed and published studies grows every week. And so on that note, I would like to introduce our first set of speakers for the discussion of prophylaxis. Dr. Stephen Quay is the president and CEO of Otosa Therapeutics. He trained as a postdoctoral fellow in the chemistry department at MIT, was a resident at the Harvard MGH Hospital, and spent almost a decade on the faculty of Stanford University School of Medicine. A TEDx talk he delivered on breast cancer prevention has been viewed over 200,000 times. His 300+ contributions to medicine have been cited over 9,900 times, placing him in the top 1% of scientists worldwide. He holds 87 US patents and has invented 7 FDA-approved pharmaceuticals, which have helped over 80 million people. He is joined by Dr. Ashish Manglik, who is an assistant professor at the University of California, San Francisco. His research laboratory normally focuses on the molecular basis of transmemory signal transduction by G protein coupled receptors, with an aim to define the atomic basis of hormone and drug reaction. Toward this goal, his lab has generated new approaches for rapid discovery of conformationally selective nanobodies from synthetic libraries, yielding an open source platform currently in use by many academic and industry laboratories. These approaches have enabled rapid discovery of SARS-CoV-2, neutralizing nanobodies with potential use as aerosolized approaches for passive immunity. And with that, Dr. Quay, please go ahead.
Thank you very much. I'm sharing my screen here. Okay, it's a pleasure to be here again at the PMWC meeting, even if it's only virtual, but hopefully next year we can be in Mountain View together. Otosa Therapeutics is a public company. And so we have our SEC filings that you should refer to. We actually have two programs, one in breast health, which I won't speak about today, where we have two phase 2 trials, one in neoadjuvant treatment, one in to reduce mammographic breast density. But two trials in COVID, one for nebulized formulations for lung recovery and our nasal spray, which I'll speak about mostly today. As a reminder, COVID has a sort of a 3-phase disease where early on it's primarily replicates in the nasopharyngeal region through days 0 through 3. If it continues, then it goes down into the upper and then eventually the lower airways days 3 through 10. And if it's not stopped there, it becomes a systemic viremia with end organ damage throughout. We believe there are 4 areas based on this disease that therapeutic intervention could be used. One is in prophylaxis and prevention, a nasal spray given at the time of diagnosis, or to people living in a household with someone early diagnosed, could help lower the disease burden or prevent it entirely. At the time of arrival in a hospital, systemic therapy is needed to prevent that viremia. We also have, think it's very important for an inhalation drug to either prevent requirement of a ventilator or to get someone off the ventilator early. And of course, now we know with long haul patients that organ recovery, especially lung recovery, I believe is a therapeutic target. So what is the role of therapeutics in 2021? I think they fall into kind of 4 categories. One is an alternative. As we're getting real world experience with the vaccines, we're beginning to see populations at high risk of severe vaccine reactions. So perhaps we may define a population where a therapeutic would be maybe more appropriate. The other is as a belts and suspenders approach. If the efficacy begins to not be as high as we think, perhaps in certain people who have very high exposure, again, first-line medical people or people in the service industry might want to use a nasal spray that would prevent infection during their 8-hour shift or something like that. Third might be a bridge to vaccine 2.0. We completely understand how this virus works, and both infections and vaccinations are going to put pressure, genetic pressure on the virus to escape. And so there will be a need at some point for a vaccine 2.0, but a bridge to that may be necessary with a nasal spray that reduces the nasal burden. And finally, again, the post-COVID recovery with up to 35% of people, or almost 28 million people, having long-term effects, we believe is an important therapeutic target. Our AT301 nasal spray program is effectively a nasal mucosa vaccine. The virus first, the spike protein of the virus first binds to ACE2, which is the very first step in the process, and then 3 host proteases are required to activate the virus for infection. ACE2 itself, furin, and TMPRSS2. Our formulation contains proprietary inhibitors of all 3 host proteases. It's intended for outpatient use. We've completed a phase 1 trial where we gave it 3 times a day, every 6 hours, 6 to 8 hours for 14 days in a phase 1 trial, and it was very well tolerated. We're now completing the data review there and preparing for a phase 2 study in the US, where patients will be newly diagnosed by PCR with COVID, and then if they have an adult living in their household, we will try to do a prevention with them as well. So with that, I'd like to turn the slide back over to Monica.
Thank you so much, Dr. Quay. Dr. Manglick, would you like to go ahead?
Hi, everyone. Thanks, Monica. Thanks to the organizers for putting on this great conference. What I'd like to tell you about today is really an academic project led out of a group that pivoted to work on COVID-19. So my lab at the University of California, San Francisco normally works on completely different topics, and we were joined by a cadre of amazing scientists across our university to focus on this problem. So the central thing that I'll tell you about today is a very alternative and different approach to providing passive immunization for the SARS coronavirus. But before I get into that, I'd like to highlight that the fundamental way that this virus gains entry into the body is pretty well characterized really after almost 2 decades of work since the original SARS virus. And that is that the spike proteins on the surface of this virus have to obligately interact with the ACE2 receptor on the surface of either the nasal passageways or in the lungs. Now, this binary interaction has been mapped to exquisite detail again, over the past 17 years, where we know the 3-dimensional structures of the spike protein and the ACE2 receptor, and we know how this interaction really happens at a deep level. And blocking this interaction, therefore, is a very potent way to prevent viral entry. And as a result, is the primary way that a lot of the vaccines that are now starting to be distributed target your own body, one's own body to induce antibodies that block this interaction. So that's one way of providing long-lasting immunity. An alternative approach that really got off the ground at the start of the pandemic was to, in parallel, look for approaches to provide passive immunity. And 2 primary ideas have really found a lot of traction. The first of which is convalescent plasma. And the general idea here is one could isolate the antibodies that are produced by a patient that's been infected with COVID and use that either in a prophylactic setting or in a therapeutic setting. I won't talk a lot about this because you'll hear quite a bit about this approach a little bit later in this session. The other approach, in contrast, is so-called monoclonal antibodies. And the idea here is that instead of isolating a panel of antibodies, what one could do is isolate a single antibody with the required characteristics, scale that up, and then give that to patients that are either sick with COVID or are high risk. And the challenge with monoclonal antibodies has been really one of both efficacy and logistics. So now a number of trials have shown that while a number of groups beelined ahead to try to develop these approaches, they really have only marginal efficacy for patients that are sick. But on top of that, because they have to be given via the blood, the logistical challenges of having patients come to infusion centers for prophylaxis is really quite hard. And on top of that, the amount of doses required, really enormous doses, about 50 milligrams per kilogram, really make it hard to have this as a widely deployable strategy. So when we approached this problem, we wanted to come at it a slightly different manner. What if we could give one of these neutralizing agents directly to the airways in a nasal spray or in an aerosol? And the key advantages of an approach like this would be that it would be self-administered by the patient, again, getting around those logistical challenges. And then because it's delivered directly to the site of infection, it could perhaps be a lot more effective. And the challenges for doing this are that one requires a really ultra-stable biologic. And on top of that, the pharmacokinetics of this kind of approach are really not well worked out. So the way that our group approached this is to identify a so-called nanobody that targets the spike protein of the SARS coronavirus. So what are nanobodies? Nanobodies are these fascinating small immunoglobulin domains that are basically derived from a part of conventional antibodies that normally exist in camelids like alpaca or llamas. So again, they're very small, ultra-stable proteins. They're relatively similar to human antibody heavy chains, so they're not recognized as foreign by the human immune system. And because they're so simple, they can be easily produced in microorganisms like yeast or E. coli and be made relatively cheaply. So what our group did is to find nanobodies that target the spike protein. And the conventional way of doing this would be to immunize an animal and go through that process. By contrast, my lab has developed a library of about 2 billion individual nanobodies, really inspired from llamas, but displayed on the surface of yeast. And then we go into this library of nanobodies to find ones that meet our criteria. For this project, looking for nanobodies that bind to the spike protein of the SARS coronavirus. And out of this campaign, we isolated one nanobody that binds with exquisite potency, femtomolar potency to the spike protein, and inhibits its interaction with ACE2. We've learned quite a bit about how this reagent works. Here's, for example, a cryo-EM structure of one of our nanobodies bound to the spike protein. And we know precisely how it sits down on the top of the spike protein and prevents its interaction with the ACE2 receptor. And we've subsequently been able to show in cell entry assays that this reagent, which we call ARNAB6, inhibits viral entry with incredibly potent doses, in this case, with an IC50 of about 50 picomolar, again, reflecting the lower limit of this assay. So with this, we're we're an academic group. We didn't really set out with a lot of necessarily industry expertise in translation. So we're now at the range where we're trying to bring this past the translational valley of death. And you can see a nice presentation or a discussion with Peter Walter, one of the key collaborators in our ARNAB team, in another session with Kiti Yamamoto, discussing some of the challenges that we face really translating this approach. So with that, again, I'd just like to highlight a really remarkable team of contributors to this project, and one of which you'll hear about in just a minute, Daniel Swaney. Thanks, Monica. Back to you.
Thank you so much, Ashish and Steve. The wonderful overview of your compounds. I guess one of the key questions would be, Stephen mentioned a little bit of what happens to vaccines when there are escape variants. So for both of your compounds, this is a question for both of you. How robust to mutational changes in the spike protein are your compounds?
Who would you like to go first?
You can go ahead.
Oh, okay. So I think these were very nicely complementary talks, because one dealt with the first interaction, spike and ACE2. Our program deals with the 3 host proteases. One of the attractions to our approach was that host protease escape, we believe, is a very distant possibility. There probably is an endosomal pathway that then would use cathepsin-like enzymes in the endosome, but from our perspective, we think that escaping host protease activation is very unlikely. I'll stop with that.
I think one of the challenges for all monoclonal antibodies or nanobody type approaches that directly target spike is that the same pressure that the antibodies, the vaccines produce at some level will happen there. I think what's clear is that having cocktails of nanobodies, and that's what we're developing in parallel, or nanobodies that target multiple epitopes, really severely decreases the probability of resistance. And on top of that, there are one of the things that we're trying to do are find nanobodies that can target other epitopes. On top of that, because we have relatively good ideas, at very high structural resolution, the specific contacts that are made, the ability to find new nanobody variants that tackle new resistance variants is actually a pretty rapid process. Now, the question of how that one translates that, across the development landscape is a different story, but actually the act of finding one that, that neutralizes a new resistance variant is almost trivial at this point.
Wonderful, thank you. And in terms of availability for your AT301 compound, Steve, what is the time frame in which you think it could be therapeutically available?
Sure. So fourth quarter last year, we finished a phase 1 trial in Australia. We're wrapping up that data, and we'll be seeking permission from the US FDA for a phase 2 trial, which again will be, the inclusion criteria will be 48 hours of a PCR positive COVID, and then if there's there'll be a prevention arm, we hope, in negotiation with the FDA, and that would involve adult patients in the same household for prevention. So we want to kick that off as quickly as we can here in 2021, with the idea of successful, perhaps seeking emergency use authorization.
Great. And similar for you, Ashish, what is next for ARNABs, and what's needed to cross this translational valley of death?
Yeah, great question, Monica. So, I think what our group, or what the academic group like us is really good at is the early phase of discovery. And I think we can be relatively innovative in finding molecules pretty rapidly. But I think the kind of more rote things like doing the IND enabling studies and teeing up to the FDA to really pass the hurdle of getting it into clinical trials is something that most academic groups, certainly ours, doesn't really have expertise in. And so we're strategically for us, we're trying to find that right group to enable us to get over that hurdle.
Well, good luck. George, do you have any? I think we have time for one more question.
Yeah, sure, please. So both of you propose, I think with the nasal application, sort of a very attractive, hope it's not a fantasy of being able to give a squirt once or a few times a day, perhaps, and to be able to actually have very strong prophylaxis. Can you give us any sense as to what's a sort of minimum or maximum duration that you think that sort of dosing would enable?
Well, I guess if I can maybe go first here. So I've invented two nasal sprays that are FDA approved, one for anemia and one for osteoporosis. So one of the challenges is that the nasal mucosa is a bit like lava flowing down a mountain. So with no adjustment to the formulations, it probably clears within 30 to 60 minutes. In the past, we've been able to get formulations that extend that to 4 to 6 or 7 hours. So in our clinical trials, we used it 3 times a day, every 8 hours as a concept for maybe perhaps a work shift, for a service provider or something like that.
Thank you.
Yeah, I can answer briefly. I'd largely defer to Stephen on this, mostly because the formulation side of it, again, we would largely hope to tap into the kinds of work that Stephen and other people who've done formulations development for intranasal or pulmonary delivery would have already figured out.
I think with that, we thank you both. That was really informative. And we move on to our next mini session. Thank you, Ashish. Thank you, Steve.
Thanks.
So we continue with our next topic, antivirals. Our speakers are Dr. Daniel Swaney, an assistant professor at the University of California, San Francisco. During her PhD training at the University of Wisconsin, she developed novel approaches to study phosphorylation signaling and leveraged these approaches during her postdoctoral training at the University of Washington to enable high throughput characterization of crosstalk between phosphorylation and ubiquitin signaling. Recently, Dr. Swaney's lab has been focused on the discovery of protein-protein interactions and phosphorylation signaling that regulate SARS-CoV-2 infection. Dr. Anu Osinusi is the Executive Director of Clinical Research leading the Emerging and Respiratory Viruses Group in the Virology Therapeutic Area at Gilead Sciences. She is an infectious diseases physician with over 15 years of experience in infectious diseases, liver disease, public health, and drug development. In her current role, she oversees development efforts for COVID-19 therapeutics, including remdesivir. She is the author of over 70 scientific publications and has a passion for rapidly advancing transformative therapies. Danielle, go ahead.
Thank you. I'm excited to be presenting today on our work studying phosphorylation signaling in SARS-CoV-2 infection. So viruses such as SARS-CoV-2 have a limited number of viral proteins with which to carry out all the functions they need for survival, and thus, upon infection, rapidly hijack host cellular processes to promote their own survival. Likewise, the host has machinery to support a robust defense to ensure its own survival. Phosphorylation signaling plays an important role in both of these processes, as the addition of a phosphate group enables the modulation of substrate function in a rapid manner and allows for the propagation of signaling downstream of a given substrate to vast networks of other substrates. This rapid nature of phosphorylation signaling is crucial to the race between the virus and the host for survival. So here we have used a proteomics approach to map how phosphorylation signaling is regulated upon infection. Specifically, we infected Vero E6 cells, which are cells derived from African green monkeys that are highly infectable by SARS-CoV-2. Upon a 24-hour time course of infection, we harvest cells and compare phosphorylation signaling to that of a mock infection. To measure phosphorylation changes, we use a proteomics approach. And using this approach, we to quantify thousands of phosphorylation sites. We find that several thousand of phosphorylation sites are differentially either up or down regulated upon SARS-CoV-2 infection. To enable the interpretation of this vast information data set with thousands of phosphorylation sites, we relied on a tool that maps these individual phosphorylation substrates to a given kinase that regulates that site. With this information, it aggregates a substrate quantification and translates this into a kinase activity information. So shown here is a subset of some of the most highly activated, as shown in red, or inactivated, as shown in blue, kinases from our data set. And these kinases represent many different classes of cellular kinases. Importantly, this translation of phosphorylation signaling information to kinases enables us to prioritize many drugs, because many of these kinases are already mapped to known drugs. So I'll briefly give an example of how we can map these kinases to drugs. So one example that we found here is the case of casein kinase 2, which is known as CK2. This is a multi-subunit kinase that we find to be rapidly increased in activity upon infection, and this increase in activity is sustained throughout the 24-hour time course that we did our measurements. From our previous papers, we had already shown that SARS-CoV-2 N protein interacts with multiple subunits of casein kinase 2, and now our phosphorylation signaling analysis reveals that downstream substrates of CK2, which are involved in cytoskeletal reorganization, are increased in phosphorylation level. From this, we observed this led us to look at some cytoskeletal features upon infection, and we found that extracellular filopodia, as shown here, were increased in frequency upon SARS-CoV-2 infection. And in fact, the SARS-CoV-2 N protein and CK2 kinase co-localize in these filopodia. Furthermore, when we use transmission electron microscopy, we can zoom in and look at these filopodia, and what we observe is viral particles budding from these filopodia. So this suggests that the activation of casein kinase 2 and its interaction with N protein may serve as a mechanism for cell-to-cell transmission of SARS-CoV-2 virus. So next we wanted to test if inhibiting casein kinase 2 could inhibit viral replication. So we mapped these casein kinase 2 subunits to a database of drugs and observed that one drug, cimetosartib, was in phase 2 clinical trials for cancer. We then performed in vitro replication assays and find that indeed this drug can inhibit SARS-CoV-2 viral replication with micromolar IC50 values. Now, CK2 is just one example of a kinase that's regulated upon infection. We mapped all of the kinases that we found regulated from this experiment to different drugs. And in total, we tested 68 of these drugs in vitro, and 38 of them we find that they have IC50 values well below their cellular toxicity. Lastly, everything I've shown you here is an in vitro assay, so the question remains, can this be translated to patient care? And so this CK2 inhibitor, cimetosartib, was recently given emergency IND authorization and has been administered to one patient. That patient was on a ventilator for 2 weeks and given a multitude of different drugs with no improvement in symptoms. And upon administration of cimetosartib, they were off the ventilator in 1 day and discharged 5 days later. So we find this to be a really promising result that hopefully can be applicable for more patients. So in summary, we use global mapping of phosphorylation changes, translate this into kinase activities, and then use this to prioritize drugs that we think could be efficacious for antiviral repurposing. With that, I'd just like to acknowledge all of the co-authors on this publication. It was an international effort between multiple different institutions across the world, and our funding and institutional support.
Thank you, Danielle. Anu, whenever you're ready.
Thank you. So thank you for the, thank you to the organizers for the invitation. And thank you, Danielle, for that excellent talk. I think it's an excellent segue into what I'll be briefly discussing today, which really revolves around developing a small molecule antiviral for COVID-19, namely remdesivir. Okay. So really, what is remdesivir? Remdesivir is the first approved therapeutic agent for the treatment of COVID-19. It's a prodrug of an adenosine nucleotide analog with broad spectrum, potent activity against multiple classes of viruses that are associated with significant morbidity and mortality, including coronaviruses. So when you look at this slide, while the primary programs were essentially for acute Ebola virus, during that time frame, we also collaborated with other researchers to study the effect of remdesivir against the other two identified human coronaviruses, namely MERS and SARS. However, despite really positive in vitro and in vivo preclinical data, remdesivir could not be advanced into clinical development for coronaviruses at that time, as there are very limited cases of pathogenic infections in humans to support the robust clinical safety and efficacy testing that you would need, and then COVID-19 happened. And so, moving on, really very early on in this outbreak, before we had the preclinical data, or before we actually knew there was human-to-human transmission, or that this would become a pandemic, we really mobilized around 3 pillars very early on in January of 2020. The first was, let's figure out as quickly as possible if remdesivir works for this disease. Second, in parallel, how do we ensure that there is adequate supply of remdesivir, which is a very complicated drug to manufacture? And third, how can we ensure that there's really responsible access? So the first pillar was really to just establish safety and efficacy by generating clinical data at a time when there was very limited information about this disease. What was the natural course? What was the progression? What are meaningful endpoints to study? What populations would you benefit? And so we had to extrapolate from other diseases, and namely, we really looked at influenza, where there's been a lot of research that has been done in that space. And this was also at a time when there was incredible logistic and operational challenges with test kit shortages and PPE shortages. And then the second pillar was really around manufacturing. So again, very early before we had preclinical data, we aggressively implemented this multi-pronged approach to manufacture remdesivir. And this was a critical decision, as production of remdesivir itself is very complicated and resource and time intensive. It involves a long linear chemical synthesis process over 20 steps, most of which have to be done in a sequential fashion that require multiple weeks. So we knew that the thing that we could not buy was time, and that we had to move really fast. And so put in place a number of process developments and refinements that took that manufacturing timeline from 12 months to down to 6 months. And so, when we look at where we were last year, we had 5,000 treatment courses, and now we're at a point where we're actually able to meet the global demand with millions of treatment courses. And then I think the third part was commitment to responsible access. Our early access programs, including compassionate use in pediatrics, pregnant women, expanded use programs, really to ensure that there was really no one left behind, and working with generic manufacturers to ensure that remdesivir was manufactured in 127 low and middle income countries, similar to what we've done with HIV and viral hepatitis. So what does the clinical data with remdesivir really show? Shown here is the NIAID Act 1 trial, which is foundational evidence supporting the use of remdesivir in patients with COVID. And so the primary endpoint of this study was really time to recovery, which means that you're well enough to leave the hospital or you return to normal activity. And so what we saw was remdesivir reduced that time to recovery by 5 days. And when you look at sicker patients, this was 7 days. And so this, we believe, is clinically meaningful, that people are able to get back home faster, back to their loved ones faster, and get out of the hospital quicker. And then just really touching really briefly on some of the secondary endpoints in this study, I think that are also meaningful for patients and providers, was looking at higher rates of clinical improvement with remdesivir, a shorter time to discharge, shorter time of initial hospital stay, and lower rates of disease progression. So 43% of patients, 43% fewer patients on remdesivir progressed to that critical part of needing mechanical ventilation or ECMO. And when we look at the fact that we're still currently in a surge pattern, having a drug that reduces healthcare resource utilization remains meaningful. And then I think the last piece is mortality. Now, this study was not powered for mortality, which we still think is an important study, an important endpoint. Nevertheless, there was a trend showing that in a post hoc analysis to figure out who responded the best, when you look at patients who did not have critical disease who are not on a ventilator, you could see a 70% reduction of mortality in that subgroup. And I think this is consistent with what Monica showed earlier, that when you treat patients early before you get to that critical stage of disease, then you have better outcomes. And this has been the body of evidence leading to approval in more than 50 countries. And then I think looking at the future and our ongoing efforts, there's still unanswered questions and areas of unmet need. We're conducting additional studies in patient groups with unmet needs, such as pediatrics, end-stage renal disease. In parallel, remdesivir has been studied in a combination approach with other targets with complementary mechanisms of action on a variety of platforms, primarily in Operation Warp Speed. And that includes JAK inhibitors, IL-6 inhibitors, IVIG, neutralizing antibodies, to see how we can combine these and optimize outcomes in these patients. And then finally, I think we're trying to look at other therapeutics that are more convenient for patients in the outpatient setting by exploring alternative formulations, such as inhaled or other non-intravenous routes of administration. So, finally, I think I'll just like to acknowledge a number of partners and collaborators that we've worked with over the years, and the thousands of researchers, and most importantly, patients that have made the story of remdesivir possible. So I think we'll hand it over back to Monica at this point.
Thank you, Anu. That was a really great overview, and it really underscores how all of industry came together to really deliver. For the sake of time, I think that maybe we only have one question, and I would like to ask that last point about alternative forms of drug delivery. What are the key challenges that you envision for, say, a nasal version of remdesivir or some of these other analogs?
Yeah, thanks, Monica. I'll keep it really brief as well. So I think, we know that remdesivir is a nucleotide, and nucleotide analogs are very notoriously difficult to make into oral compounds because of the metabolic clearance and bioavailability. So we've had challenges, but we've been able to do it in the past. So I think, we recognize that oral agents are probably, or other routes of administration are probably more convenient and feasible for patients, and it's something that we're pursuing. But nucleotide analogs as a class of drugs are notoriously difficult to make into oral formulations.
Thank you. And I know that we remain with many questions, but I really want to thank you both for the great overview. And with that, then I will introduce our next speakers. So we move on to our discussion on convalescent plasma with Dr. Aaron Tobian and Dr. Peter Marks. Dr. Tobian is a professor of pathology, medicine, and epidemiology at the Johns Hopkins University School of Medicine and Bloomberg School of Public Health, and the director of the Transfusion Medicine Division at Johns Hopkins Hospital. He sits on the editorial board for the journal Transfusion and is the founding editor-in-chief of the website Transfusion News. He has served on the American Association of Blood Banks Board of Directors for the past 4 years and was just elected to join the executive committee of the board of directors. He also has an established global research program that studies transfusion-transmitted infections, and more recently, the use of convalescent plasma to treat or prevent COVID-19. Dr. Peter Marks received his graduate and medical degrees from New York University and served as clinical director of hematology at Brigham and Women's Hospital in Boston. He then moved on to work for several years in the pharmaceutical industry prior to returning to academic medicine at Yale University, where he served as chief clinical officer of Smilow Cancer Hospital. He joined the FDA in 2012 as deputy director for the Center for Biologics Evaluation and Research and became center director in January 2016. Dr. Marks and center staff are committed to facilitating the development of biological products and providing oversight throughout the product lifecycle. We're delighted to have you. Aaron, please go ahead.
Thank you very much for the invitation to speak today. It's a pleasure to talk about convalescent plasma for the prevention of SARS-CoV-2 and treatment of COVID-19. And in March, as the COVID-19 pandemic began to reach the United States, our group had proposed that we should consider convalescent plasma. Immune or convalescent plasma refers to plasma that's collected from individuals following resolution of infection and development of antibodies. Passive antibody therapy through the transfusion of convalescent plasma may prevent clinical infection or blunt clinical severity in individuals with recent pathogen exposure. Unlike many of my colleagues who are recently talking about new techniques, convalescent plasma or passive antibody therapy has been used for over a century, first with the influenza pandemic in 1918 for treatment. It's also been used for Argentine hemorrhagic fever, SARS-CoV-1, Ebola, and Middle East respiratory syndrome. In addition to treatment, convalescent plasma can be used for post-exposure prophylaxis. Thus, people who are exposed but not yet infected with hepatitis, mumps, polio, measles, rabies. And passive antibody therapy is most effective when administered early after the onset of symptoms or administered prophylactically. We thought convalescent plasma would be the best and only short-term strategy to begin with COVID-19. Early in the pandemic, we worked to create our own repository of donors since convalescent plasma was not yet available. What we found is among these donors who were approximately 40 days following a positive PCR test for SARS-CoV-2 is that there were diverse antibody isotypes that were detectable, and IgG is the prominent isotype found across a diverse assays and analyses. We also found there's a large heterogeneity in the antibody response that's consistent with previous studies. We found about 10% of individuals did not even have detectable neutralizing antibodies. There was a group that had a medium level of antibody titers, and then others that were very high titer. In addition, we found that neutralizing antibody titers were higher in those who were male, those at increased age, and those who were hospitalized. The antibody titer levels appeared to be consistent with those who had significant and severe COVID-19 disease. Antibody levels were also constant, but decreased over time. So the ideal time to collect convalescent plasma was between 3 and 9 weeks post-infection. In terms of how convalescent plasma might work, most people focus on the neutralizing antibody activity. And these titers are thought to decrease viral load and decrease inflammation. However, our work and others have also shown that in addition to the neutralizing antibody activity, that antibodies may mediate other pathways such as complement activation, antibody-dependent cellular cytotoxicity, and also phagocytosis may contribute to the therapeutic effect of decreasing respiratory demand, increasing radiologic improvement, and then overall improving survival. More than 100 randomized clinical trials of convalescent plasma were initiated, and we're just now beginning to see some of the data. Some of the meta-analyses are beginning to show benefit. It's unclear when you combine all the trials and observational studies of convalescent plasma who are benefited. However, it's important to look at specific patient populations. Among those individuals who appear to work best, it's the post-exposure prophylaxis to prevent infection, those with early treatment to prevent hospitalization, and treatment soon after hospitalization. Those who are severely ill or those who are already intubated do not appear to benefit as much for convalescent plasma. Other work's being done to look at pediatric patients and those who are immunosuppressed. So in conclusion, convalescent plasma has been used for over 100 years as prophylaxis and treatment. COVID-19 convalescent plasma decreases viral load and inflammation. Current trials and observational studies suggest COVID-19 convalescent plasma may be effective, especially among those receiving it early. But it's most important to continue to pursue the randomized trials and explore the FDA's expanded access program to understand the full efficacy profile and the best patient populations. Thank you very much.
Thanks very much. So I'm going to talk about the COVID-19 convalescent plasma expanded access program that was run in the United States. And This was a protocol, which was a single-arm protocol, which was really put in place to provide access to convalescent plasma at community sites across the country at a time when the COVID-19
Thank you.
pandemic was really
That was great.
growing
And that's
very quickly.
so nicely for what.
At that time, there were a large number of cases increasingly coming up in the country, and we needed to have a way to get access. There were a number of demographic and simple endpoints that were captured as part of this protocol that included safety, 7 and 20 day outcomes. And a key feature of this was by the time that August had come around, after this was started in April, there were over 100,000 individuals enrolled and well over 70,000 people treated. Now, one of the important things to note in this trial was that during the course of the period between April and August, there were a variety of treatments that came on the scene that subsequently came and went. For instance, hydroxychloroquine was there at the beginning and kind of went away during the middle, and dexamethasone started to come in, and well, the monoclonal antibodies. So there were a variety of concomitant medications that were used, and those are hard to correct for as part of this trial. Nonetheless, it wasn't really a trial, it was a study, I should say. Some findings that we had were that the accurate determination of the antibody titer was really critical here, and it took us a while to get this right. A comparative study that we conducted in June determined that several of the tests we were using to determine antibody titers were unreliable, and we ultimately settled on the Broad MIT BSL-3 neutralization assay, which proved to be quite reliable and almost a reference standard. It was a live virus neutralization assay. The range of the antibody titers in the Broad assay was spread across a very wide range from essentially no antibody detected to very high titers. And there was a relatively even distribution of titers across the range. Just to show you some of the data from this study, the effect sizes were modest. When one looked at 7 days in individuals who were hospitalized for 7 days, or who died prior to day 7 or on day 7, and one looked also at day 28 and individuals who had been hospitalized for up to 28 days for the 28 days, or who had died before 28 days or on day 28, one could see that there were modest improvements in those who received high titer plasma compared to those who received lower titer plasma. There's no control arm here, obviously, because everyone received plasma. But when one compares those who received higher titer plasma, that is plasma that had, in this case, levels of over 250 in the Broad assay, one saw a very modest reduction in the overall population of about 1.5%, with a relative improvement of about 10% that was statistically significant. If one really honed in on those who really benefited the most, it was those who were not intubated, where there was about a 15% relative improvement, 2% absolute improvement. And that was at day 7. And those also carried out to day 28. Now, one should note that by day 28, the mortality is pretty high, because if you were in the hospital for 28 days with COVID-19, it means you had a pretty protracted hospital course. This is just to show you these plotted by the titers themselves. And you can see this cut point of 250 compared, particularly on the orange bars here, you see that those who were not intubated, who received higher titer plasma, did better compared to those who received lower titer plasma. And it did appear to be some dose response here, although again, it's modest. So that carries over to a Kaplan-Meier plot analysis here, where you can see there's an effect in the overall population, but it's really driven by the effect of those treated early who were not intubated. And again, although the p-values here are very, very small, it's because the number of patients here, and numbered here, as you can see, 10,000 or more, is large. So that's how these small these small differences turn out to be highly statistically significant. And one could argue that 10 or 15% relative improvements here, or 2% absolute improvements, are meaningful when you think about lots of individuals. So takeaway messages here are that high titer versus low titer convalescent plasma is associated with a modest survival benefit that was most apparent earlier in the disease course. Overall, about a 1.5% absolute improvement, 10% relative improvement at 7 days. And for those who weren't intubated, it was about 1.9% absolute and a 14.5% relative improvement.
Thank you so much, Peter. That was great. I had a bit of some technical difficulties, but I'm back. And since George didn't get to ask any questions the last session, George, I'm sure that you'll have questions for both Peter and Aaron.
Sure, thank you very much, Monica, and thanks to both Aaron and Peter. One question that strikes me about the convalescent plasma, Aaron, you indicated that it has sort of multiple mechanisms through which it can act. What I'm sort of wondering is if there's a specific class of patients that might most potentially benefit from convalescent plasma. I'm thinking for one, potentially immunocompromised individuals who may not respond well to a vaccine. I'm wondering if this could be a particularly good subgroup to hone in on.
So it appears one of the clear messages is the earlier the treatment with convalescent plasma, the better. And I think, as you point out, George, those who are immunocompromised are a group of real interest. And at our hospital, we're occasionally treating those who are immunocompromised, who are coming in without severe disease immediately with convalescent plasma. And I think we need additional studies to really assess the efficacy, but that is one area of significant interest.
And Peter?
Yeah, I couldn't agree more. And I think if you assemble the data, for instance, on immunocompromised individuals, again, it's not a randomized trial. It's not even a well-conducted trial. But if you collect these case series that are small, it does look, particularly in immunocompromised individuals, we're seeing it particularly in hematologic malignancies patients, where there are now a couple of papers that have appeared of case series of somewhere between 5, 10, sometimes a little more patients, that they does seem to have benefited those individuals significantly, even if they weren't treated quite as early in the course. That said, getting back to what was said by Aaron, that really on the whole, people should be treated early. It looks like early is better here. And toward that end, there are randomized trials going on treating very early, that is as outpatients, and hopefully those will bring us the kind of data that we need to be able to rely on.
And maybe just a quick follow-up. Can you contrast the use of convalescent plasma potentially to monoclonal antibodies, whether cocktails or single agents, whether inherently convalescent plasma might have any advantage or they're the converse?
Well, I may start by just saying that I think the advantage of convalescent plasma, if it's used early, first of all, it's similar. What we're seeing is it's very similar to monoclonals. The earlier you use it, the better. But to the extent that we're starting to see variants pop up, the nice thing about polyclonal responses is that there's more likelihood that there will not be an escape from some version of the virus.
Excellent point. Monica, how are we doing on time?
I think that we could stay here for a long time, but we're going to move on. Thank you, Peter. Thank you, Aaron. And I'll introduce our next speakers. So we will finalize the session by discussing anti-inflammatory therapies with our speakers, Dr. Max Koenig, a fellow in clinical and molecular rheumatology at the Johns Hopkins University School of Medicine. He has a long-standing interest in mechanisms of disease initiation and propagation in autoimmune, rheumatic, and inflammatory disease. At the Ludwig Center at the Johns Hopkins University, he is part of an interdisciplinary team of physicians and scientists pursuing clinical trials to prevent the development cytokine storm syndrome in COVID-19 by disrupting self-amplifying catecholamine cytokine signaling pathways. And Dr. Bruce Patterson, the CEO of IncelDX. During the early stages of the AIDS epidemic, Dr. Patterson began investigating cellular reservoirs of HIV-1 using molecular and in situ cell-based technology patented in his laboratory and used today around the world. Dr. Patterson determined that enough HIV virus was present in infected individuals to account for the massive destruction of the immune system. He has authored over 150 manuscript and book chapters focusing on single-cell biology and diagnostics, and was formerly Associate Professor of Pathology and Infectious Diseases and Director of Virology at Stanford University. He has translated his research discoveries into single-cell diagnostics for cancer, immuno-oncology, and infectious diseases. Max, go ahead.
Well, thank you very much, Monica. Thank you, George, for allowing me to join such a terrific group of speakers today. Really excited. So on behalf of the Ludwig Center at Johns Hopkins, it's my great pleasure to discuss our approach to prevent death in patients with COVID-19 using alpha blockers. Undoubtedly, the vaccines are the success story of this pandemic, but as we heard, they're not 100% efficacious, and as was already alluded to, they may not be as effective against all emerging mutant virus strains that we're currently encountering and may identify in months and years to come. So inevitably, people will still get infected and people will still die, and we need additional approaches. There's considerable evidence at this point that in the most severe cases, COVID-19 associated hyperinflammation, which really is a dysregulated immune response against the virus, is a primary driver of organ damage and mortality. Unfortunately, most efforts to repurpose drugs for their presumed antiviral activity has failed during this pandemic, and we lack therapies to effectively treat hyperinflammation when end organ damage has already developed. As you know, dexamethasone is one of the few examples that has made any dent in mortality in this setting. So early in this pandemic, we proposed a completely different approach to all these immunosuppressive strategies in COVID-19. We believe that we really need to prevent hyperinflammation in patients with early disease rather than trying to treat end organ damage that has already occurred. The analogy is obviously that if the house has already burned down, there's only so much water can do. So we think that alpha-1 adrenergic receptor antagonists, I'll call them alpha blockers now, are a very promising class of drugs to prevent hyperinflammation in COVID-19. It may be slightly counterintuitive. So alpha blockers like doxazosin, prazosin, tamsulosin are a class of drugs that have been widely used for decades in the treatment of prostatic hyperplasia, hypertension, PTSD, are safe, and importantly, are not immunosuppressive. Unlikely that they interfere with antiviral immunity. Our proposal was based on recent preclinical data from our group, which showed that alpha blockers can prevent cytokine storm syndromes and death in animal models. The established mechanism identified is that these drugs are able to interrupt the positive feedback loop between catecholamine signaling and overproduction of cytokines, as highlighted in the different graphs in the top of the slide. As you see in the bottom left, doxazosin, which can block all three alpha-1 receptors, but not tamsulosin, was able to rescue mice with hyperinflammation and prevent death. We therefore wondered whether this mechanism was applicable to hyperinflammation in COVID-19. And since there were no clinical trials, we conducted several large retrospective analyses of outcomes in patients with similar diseases, pneumonia, acute respiratory distress syndromes, and patients with COVID-19 who were taking alpha blockers for completely different reasons at baseline and looked at their outcomes. And I'll share just two of those. We found, for example, that in hospitalized patients with ARDS, over 100,000 patients here with ARDS or pneumonia, baseline use of alpha blockers was associated with markedly reduced mortality, an odds ratio of 0.41 in adjusted analyses. The signal here was mostly driven by tamsulosin, which may be much less efficacious than prazosin or doxazosin, which blocks all 3 alpha receptors. Similarly, in a VA cohort of 16,000 patients with suspected COVID-19, suspected here because not all patients at the beginning of the pandemic had positive testing available, we found that alpha blockers were similarly associated with reduced mortality. And here we were able to see that expectedly, doxazosin was associated with even better outcomes than tamsulosin, with an odds ratio of death of 0.38. We think that all these data still significantly underestimate the true effect size, because we only captured patients in these analyses that had progressed to a severity to need to be hospitalized. So basically, they had progressed despite the use of alpha blockers. We think that alpha blockers overall are ideal drugs in this setting because they're oral, they do not require refrigeration, they're cheap, they're readily available worldwide, and they have a very well-known safety profile for decades. But clinical trials have been lacking. So in our experience, it is very challenging to fund these trials, especially using traditional channels. So we're now planning to start a large outpatient study of doxazosin compared to placebo across the United States, with the goal to prevent hospitalization in COVID-19, severe COVID-19. And we really think we need a big enough study to answer this question definitively at a time where there is increasing pressure with changing mutations in COVID-19 and a large population that's not vaccinated. So if anybody is willing to help us fund this proposal, please don't feel in any way limited in reaching out. So this was a huge group effort. I mentioned a lot of people in the Ludwig Center at Johns Hopkins were heavily involved with this, including Dr. Chetan Betagowda, Dr. Burke Vogelstein, Dr. Joshua Vogelstein, many other collaborators at Johns Hopkins, at Stanford University, the VA health system, and other institutions worldwide. So I'd like to thank all of them. I'm only a minimal part of this team, and I'm happy to take any questions.
Thank you, Max. That was great. We'll move on with Bruce, and then we'll have our questions.
Thank you, Monica, and thank you, the organizers of the meeting. I'm here to talk about the CCL5, Ccr5 pathway in COVID. In the middle of January of last year, I was in China speaking to colleagues about this new emerging pathogen and the havoc that it wreaked upon the immune system and the cytokine system. We took samples back from that experience, and we looked at various COVID samples and found extremely high levels of CCL5 and extremely high levels of CCR5 expressed on a variety of different immune cells. As you know, and has been shown in the course of this talk and others, the COVID is really several different phases. The viral phase in the beginning, the inflammatory or immune phase, and then now we have a phase 4, which is the long haul, which I'll talk about in a moment. But I think it's critical and well within the mandate of precision medicine to understand exactly where these patients are in this spectrum of disease while we're treating them. As others have mentioned, the antivirals may not work at the end or in the immune phase of the disease. Conversely, it doesn't make any sense to give anti-inflammatories, immune modulators, or immunosuppressants in the early virologic phase. It doesn't. And that's where the meshing of pharmacology and therapeutics and diagnostics needs to take place in this very complicated disease. The mechanism of action of CCR5 antagonism in COVID really arose out of some of the studies that we've been doing in cancer. That is that CCR5 antagonism repolarizes pro-inflammatory macrophages. It inhibits the Treg effects on innate and adaptive immunity. And indeed, lately, there's some reports of possible inhibition of cell-cell viral transmission of COVID by Maraviroc a FDA-approved CCR5 inhibitor. The other thing that struck us is what you see below, and that is the level of CCR5 expression on the various immune cell types: CD14 monocytes, CD14, CD16 circulating macrophages, CD4, CD45-RO, HLA-DR positive activated memory T cells, and of course, FOXP3, CD4 positive T regulatory cells. And it's the ability to use assays such as receptor occupancy to really manipulate the system that you see in front of you and manipulate the immune cells that you see so that you favor the cells that are important, the CD4, CD45-RO positive T cells, while inhibiting the FOXP3 positive CD4 T cells and inhibiting the pro-inflammatory macrophages, which are the CD14, CD16 cells that you see below. In a study using a CCR5 inhibitor called leronlimab, which is a humanized monoclonal antibody, we saw very profound effects in early EIND patients. We saw a dramatic decrease in IL-6, which you see in the upper left, as early as day 3, and certainly very profoundly by day 14. Similarly, we saw the percent CD8 cells increase over 14 days. And I would say, similar to CD4 counts in HIV/AIDS, the CD8 percentages were extremely low in COVID, making these individuals just as immunosuppressed as you would see in HIV infection. And then, of course, when you added drug, you saw improvement in CD8 percentages. You saw a decrease of CD4 to CD8 ratios back to normal. You saw IL-6 decreasing back to normal. And all of this was because, as you see in D&E, that receptor occupancy by the CCR5 antagonists increased over time, and that was concomitant with the decrease in IL-6 and the increase in CD8s. All told, in the lower row, which we did in collaboration with our chairperson, Monica Herrera, we saw a decrease in SARS-CoV-2 plasma viral load in these severe to critical patients. And the fact that that's not being used to monitor severe to critical patients these days in response to therapy is a big question in my mind that still baffles me. This is another graph showing that SARS-CoV-2 plasma viral load obviously was most prevalent and highest in severe critical patients, less prevalent and to a lesser extent in mild to moderate patients. So it is an effective marker to monitor the efficacy of drugs in severe to critical individuals. We also saw improvements in T cell exhaustion as seen with decreases in PD-1, CTLA-4, and LAG-3 expression on CD4 and CD8 T cells. We saw repolarization of macrophages from an M2 phenotype, which is pro-inflammatory, making high levels of IL-6, to more of an M1 phenotype or effector phenotype that can help remove debris and virally infected cells in the lung. We saw the same thing when we did single cell transcriptomics. We saw, again, in the myeloid cells in the upper left-hand corner, significant decrease in interleukin-6 in the myeloid population. And similarly, in the lower right-hand corner, saw an increase in granzyme A in the CD8 component, indicative of increasing the effector component of CD8s in these individuals. All told, this led to a significant improvement in these individuals in severe critical, in EIND patients, patients on ECMO walking out of the hospital by day 5. We see with both leronlimab and maraviroc improvement by day 3, and these patients being discharged by day 5. The leronlimab trial just completed its enrollment in phase 3, and we're awaiting unblinding. Now, one thing that has come out of the COVID infection is obviously these long haulers. So we did a 4-month study of the immunity in long haulers and compared those to COVID patients. As you know, long haul patients have these significant symptoms of fatigue, brain fog, neuropathy, shortness of breath. And we developed the severity score for active COVID and a long hauler index for long haulers. And we show in this graph that long haulers and active COVID are completely different immunologically. As been mentioned, in active COVID, the immune response is chaotic and dysfunctional. However, in long haulers, they have a very stereotypical antiviral immune response with high type 1 cytokines. And that's indicative in the long hauler index of interferon gamma plus IL-2 divided by CCL4 chemokine. So we can use these indices to monitor therapy in both active COVID and in long haulers as we improve these conditions. So in summary, I mean, virologic and immunologic monitoring are critical elements of patient management now that these new therapeutic modalities are coming into play. We have to combine diagnostics for determining when to initiate certain therapies and when to stop certain therapies. The immune profiling cytokine storm assessment is critical as soon as symptoms develop, since symptoms are indicative of increased cytokines. The long hauler index and severity score that we've developed can really help triage active COVID patients into certain therapies and to monitor the efficacy of therapies in both active COVID and long haulers. And with long haulers being maybe as high as 30% of all COVID infections, despite the activity of the vaccines, we're going to be dealing with the long-term sequelae of COVID-19 for months, if not many years to come. And we have to address that with a combination of precision diagnostics and therapeutics. Thank you very much for your time.
Thank you, Bruce. That was a really great way to end this session. I have a question, and Max, actually, and it's based on these last points that you're making on long haulers. Max, do you think that with the therapies such as dexamethasone, we are inducing these patients that have viral reservoirs that then make them be long haulers? And what are your thoughts on that? And how does that change your perspective in terms of therapy?
This may be a better question for Bruce, frankly. But so the answer is I don't know. I don't think nobody knows for sure. There's certainly concern that immunosuppression early on in the disease phase might not only not be helpful, but might be detrimental, right? Just because there is theoretic concern that potent immunosuppression early on in the disease phase, the first 2 weeks before really people go into this hyperinflammatory drive, if they develop it, might be detrimental because you interfere with antiviral immunity. And even though dexamethasone has been shown to reduce mortality in people who have already oxygen dependence, especially people who are intubated, that signal was not seen in people who did not require oxygen, who probably are earlier in the disease course. And if anything, the signal was going towards maybe even detrimental. And I think similar patterns have emerged also from the rheumatic disease literature, where patients on high doses of prednisone have been treated for baseline rheumatic diseases and then developed COVID. So there's certainly concern that something like that could happen. I'm curious what Bruce thinks.
Yes, Bruce.
Mute myself.
There you go.
Great. Again, I think that's why the CCR5 antagonists are as immune modulators as opposed to immune suppressants are key to treating this disease. We are running the fine line between viral replication and immune chaos and immune damage. And the fact is, we have to be able to temper the immune damage without increasing bioreplication. And once again, I think that's what's so critical about using diagnostics appropriately in these diseases, and not just looking at O2 levels on a ventilator or on our settings on ECMO. And I think we're getting to that point. But yes, and again, in collaboration with Monica and Monica's group, we've been able to identify these long-lived reservoirs in monocytes and macrophages, much like we saw in HIV, that harbor COVID nucleic acids and even COVID protein. Whether or not it's replication competent, we don't know, but it is in fact there and can provide the continuous antigenic stimulation that we're seeing in the long haulers. And yes, I am concerned that that is being allowed to happen by the use of immunosuppressives. And I think searching for the reasons why such a large group develop long hauler symptoms is absolutely critical, because this is a terrible, terrible condition that is resource intensive, and it's going to be the leftovers after we find effective vaccines.
Definitely. I think a study correlating treatment and whether a patient becomes a long hauler or not is certainly warranted. Yeah. We are on time, perhaps a little bit over time. You have a question, George. I'm going to let you ask a question.
Yeah, please. If we could just quickly step back and ask the question about how all these different therapies fit together. There's been an emphasis on the early phase of viral replication and blocking it, later phase of tackling or preventing the hyperinflammatory. And given the concerns about, say, giving steroids, blunt steroids such as dexamethasone too early on, as we were just alluding to, Bruce, I'm wondering if you can envision, based on present knowledge, what might be the potentially sort of the ideal complementary use of, say, antivirals and hyperinflammatory inhibitors.
Again, the technology is in our hands, as we just showed. Plasma viral load is necessary. Any viral load is necessary to show when do we stop having viral replication, or are we giving dexamethasone when there's still very, very active viral replication? Conversely, are we giving immune modulators and immunosuppressives when the cytokines aren't even elevated? So the information is at our hands, and in managing patients over the holidays, the thing that was really disappointing for me was seeing how little information is available to clinicians who are managing these patients. They're solely going by clinical course and settings on the various, like I said, respirators and ECMO, instead of saying, what's the immunology doing right now? And where are we in the spectrum of disease? And we have those tools, and we need to use those tools so that we see, like a severity index, for instance, could be used to say, of these people who are coming along behind these individuals who are intubated, what's their status? Are they actually more severe than we think they are because of their oxygen levels? We have the tools. This is precision medicine. This is this conference. This is exactly what we have to do for this very, very complicated disease, is mesh therapeutics with diagnostics to know where we stand.
And yet, Max, you indicated, I think, in your studies that people that have taken these alpha blockers potentially weeks to months before they maybe contracted COVID. Is that a fair inference? That they nonetheless get significant benefit? If that's the case, might they do even better if they didn't have it too early on?
Yeah, no, it's a very good point that you're making, George. Alpha blockers are just like a blood pressure medication, right? People take them for years on end. I cannot say in these studies, actually, whether the patient was still taking the drug the day before he went into hospital or the day that he got COVID. I assume we make that assumption more likely there is a significant subset of those patients that have not been exposed to the drug. So effect sizes may be larger. Right. The point is that, you know, because people are using these drugs on a daily basis, these are not dangerous drugs, right? These are very well tolerated, and we know of decades of data that they're not immunosuppressive. They have the unique capability to modulate the immune system at that critical time that we really don't understand yet, where there is a catecholamine surge that seems to precede the development of hyperinflammation. Nobody has really studied that phase. We don't know anything about catecholamine biomarkers early on in the disease of COVID-19 that might even be predictive of people who go on to develop hyperinflammation or not. That data is not available. I think that void needs to be filled. But from a practical standpoint, a drug that has the potential to prevent that dysregulation just by modulating catecholamine signaling without interfering with antiviral immunity, right? We know people have not an increased risk of viral infection on these drugs. I think it's key. I think we just need to answer this question as soon as possible. And you know, I think all hands on deck, right? Our group has nothing to gain from it. We just want to move forward to fill that void, right? And we hope that we can do it.
You need to fund your study.
Exactly.
Yes, please fund. It's hard, right? I think we heard it. There are 100 studies on plasma. There are 200 of studies worldwide that studied hydroxychloroquine early on. And now we're in a phase where, frankly, a trial that is investigator initiated can no longer compete with industry-funded trials, right? We don't have the means. I'm not making money out of that. We don't have the means to pay $10,000 per patient, right, to recruit them. We just want to help people. So if people have money to fund, reach out, please.
How quickly do you think you could complete that study if people committed today?
If we have the money now, I think we can complete it in 2 months.
All right, everybody heard that.
Yep.
Put up your wallets.
Exactly. Well, thank you so much. I want to close this session. I want to thank all of our speakers again. I think that this has been really informative, and we could go in depth for each of these sessions. So hopefully what we've created here is just the awareness, the resources, and for people to be educated and curious for more information. Thank you again, everyone, and please stay safe.